In the following lecture, I'm going to discuss the F techniques. So we're going to focus on 4 F techniques: frap, flip, frat, and flim. So let's first start from frap. So frap, F-R-A-P, stands for fluorescence recovery after photobleaching. So now let's use an example to show you how this works. So imagine we have a membrane lipid bilayer, a 2-dimensional surface. So this is membrane lipid bilayer. So this is one leaflet, this is another leaflet, this is the side view, and this is the in-phase view from the top view. So these lipids are labeled by a red fluorescence. So it's homogeneous because lipids could diffuse freely within their leaflet 2-dimensionally. So imagine we have a circle here. This is a region of interest. This region of interest, generally in short we call it ROI. Here is a circle. So now let's imagine we have a very good laser scanning confocal microscope, and it is equipped with very high power laser. We can use this high power laser to scan this area, scan this ROI. So recall from our previous lecture that laser scanning confocal microscope is able to scan a random pattern. So if we use very high laser to repeatedly scan this area very quickly, you can imagine that the molecules, fluorescence molecules within, will be photobleached. So you'll get here. So this is done in a very short time, and usually it takes very short time. It is very possible because laser scanning confocal generally could be equipped with very high power laser. So this is from the side view. You can see that within this region, the red fluorescence molecules become photobleached, and they appear dark because they are photobleached, while the outside lipid molecules are still fluorescent. But we know that fluorescence molecules, we know that lipid molecules could diffuse freely within their 2-dimensional surface. So immediately after photobleaching, these fluorescent molecules are diffusing in, while these dark molecules are diffusing out. So by the way, we have to take note that except the fluorescence property, so here these molecules are non-fluorescent, these molecules are fluorescent, but the two populations of molecules have exactly the same physical and chemical property, except one is fluorescent, the other one is non-fluorescent. So they diffuse and eventually will reach equilibrium. So it's homogeneous. So take note that because the contribution of these photobleached molecules, so eventually, once reaching new equilibrium, the fluorescence intensity is lower than before photobleaching. So frap is usually done in the time-lapse fashion. So here is a intensity versus time plot. So this intensity is monitoring the intensity within this ROI. So this is before photobleaching. So you can see that the intensity remains constant, assuming that photobleaching is negligible. So here is photobleaching. So within very, very short time, these ROIs molecule are killed, are photobleached. So the intensity is dropped down to 0 here. Then, after the photobleaching, the fluorescence will recover. Recovery is by the free diffusion of fluorescent molecule to this ROI region. So as more molecule diffuses in, if we continuously monitor within this region the total fluorescence, the fluorescence will become higher and higher, eventually reach a plateau. So this is a recurring curve. This is the plateau. So we can see that we can use frap to quantitatively measure the kinetics of diffusion of molecules in living cell. So another application, or more general application of frap, is qualitative. So we could use it to qualitatively determine the continuity of membrane organelles. So later I'm going to give you one example. So FRAP is generally performed by laser scanning confocal microscope. This is because, as we discussed before, laser scanning confocal is able to randomly do the scanning pattern. And take note that the fluorescence recovery is always at equilibrium condition. Here is one good example for the qualitative application of FRAP in cell biology. So in the 1990s, there was a debate on the status of mitotic ER. So mitotic ER is also the ER during the cell division. So we know that during cell division, the mother cell divided into two identical daughter cells. So mammalian cells must determine how to divide everything into half, including the ER. We know that during interphase, there is only one continuous ER. So there used to be two models in the 1990s on the mitotic ER. The first model proposes there is one continuous ER. There is only one ER, and similar to the interface of ER, it is continuous. So during mitosis, there is a continuous single ER. Another model proposes during mitosis, ER disassembles into numerous small vesicles. So it sounds more reasonable for the discontinuous mitotic ER model. This is because when the ER disassembles into many vesicles, it is easier for the cell to divide the ER into two parts, to divide ER equally into two daughter cells. However, using FRAP, it has been shown that the mitotic ER is continuous. So here is the experiment. So here is the ER is labeled using lamin B receptor fused to GAP. So lamin B receptor is inner nuclear membrane protein, but here it could be also used as an ER marker. So during interphase, so here is the nuclear, and here is ER. When this ROI is photobleached, we can see very quickly, just within a couple of seconds, the photobleached ER is recovered. So FRAP recovery is really fast. This is because during interphase, the ER is continuous. Well, this membrane protein could easily diffuse from other part of ER to this ROI region. During prometaphase, so this is one of stage of mitosis. So this is the ER, this is mitotic ER. And if we photobleach this region, we can see this ROI region recovers with almost similar kinetics as the interface. So this qualitatively shows that the ER during mitosis is still continuous. So if there were vesicles, so these vesicles diffusion will be much slower than two-dimensional diffusion of membrane protein along continuous ER membrane. So if mitotic ER is numerous vesicles, so we will not have this fast kinetics of FRAP. Now let's look at FLIP. FLIP stands for fluorescence loss in photobleaching. So it is very helpful if we can place FLIP and FRAP side by side for the comparison. So here is a FLIP. So here is a FRAP. So this is a fluorescent cells is labeled by green fluor, for example, labeled by GAP tagged protein. So for FLIP, we photobleach one area, well, we monitor the other area, which is different from the photobleached area. So this is the ROI we observe or monitor, and this is the ROI we do photobleaching. So by the way, similar to FRAP, FLIP is also performed by using laser scanning confocal microscope. So we keep on photobleaching this region. At the same time, simultaneously we monitor this region's image. We can continuously imaging the whole cell and monitor the intensity within this ROI. So if the GAP molecules are all connected, all continuous, you can imagine that if you keep on photobleaching this region, so you're going to deplete the whole cell's GAP tagged molecules. So this ROI's intensity will gradually reduce. So as reduce, eventually it will become totally dark. So this is different from FRAP. During FRAP, the ROI is the region for photobleaching, and its ROI is also the region for continuous monitoring, its recovery, and its intensity. So here is a flip curve. So the ROI at the beginning, before photobleaching, is constant. After photobleaching of this region, we can see that its intensity gradually reduced. Well, for the FRAP, we photobleach a very small region. When we look at its fluorescence recovery at this region, the fluorescence will gradually recover until reach plateau. So here is an example. This is the same example to answer the question whether mitotic ER is continuous or not. So we could use flip to demonstrate that mitotic ER is continuous. So this is the mitotic ER, the laminin B receptor GIP. And this is the ROI for us to continuously photobleaching. So this is photobleaching ROI. We continuously photobleaching this region. We can see that the region that is far away from this flip photobleaching region its intensity gradually reduces. So as you further photobleach this region, we can see this intensity of the other regions reducing and reducing. Eventually, the total cells pool of laminin B receptor GIP are all photobleached. So this indirectly shows that mitotic ER is continuous. Now let's look at a FRAT. So FRAT stands for fluorescence resonance energy transfer. Sometimes it is also called Foster resonance energy transfer. So it is a type of radiationless energy transfer between two fluorophores. So one fluorophore is called donor, and the other fluorophore is called acceptor. So FRAT always involves in two fluorophores, donor fluorophore and acceptor fluorophore. And the energy transfer is direct from donor to acceptor without involving the photons or other type of energy. So now let's look at an example here. Here is two fluorophores, Psi3 and Psi5. Both are small molecular organic dyes. So Psi3 here serves as FRAT donor, Psi5 serves as acceptor. So here is Psi3's spectrum, and here is a Psi5 spectrum. So the excitation, emission, excitation, and emission. So for FRAT to occur, it requires three requirements. So the first, the donor and acceptor molecules must be close enough to each other. So the distance must be less than 10 nanometers. Generally is within 1 to 10 nanometers. So this is a distance criteria or distance requirement. The second is the spectral requirement. That is, the absorption spectrum of the acceptor must overlap the fluorescence emission spectrum of the donor. So in this case, we can see that the emission spectrum of Psi3, which is this red line here, overlaps with the acceptors. Psi5 is acceptor's excitation spectrum. So the donor's emission spectrum must overlap with the excitation spectrum of acceptor. So you can see here overlap region is here. And the last requirement is that the donor and acceptor transition dipole orientations must be approximately parallel. So this is the dipole requirement. So only if all these three requirements are fulfilled, could there be a frat between donor and acceptor fluorophore. Let's look at this example. So here is a Psi 3 labeled oligonucleotide, and here is Psi 5 labeled oligonucleotide. In a solution phase, when there is no hybridization between this oligonucleotide, if you look at this oligonucleotide, you can find that they're complementary to each other. However, without hybridization between this oligo and this oligo, this Psi 3 and Psi 5 are freely in a solution. They are far away from each other, and generally their distance is more than 10 nanometer. There will be no frat between Psi 3 and Psi 5. So now if we shine 540 nanometer excitation light, so Psi 3 is going to be excited here to generate emission light. So here 540 nanometer is around here. So its emission will be roughly here 490 nanometer. So there will be 400 nanometer emission. Well, Psi 5 will also be shined by the 540 nanometer excitation light. However, if you look at Psi 5's excitation curve, so here is an excitation curve of Psi 5 at 540 nanometer region, you can see its absorption or excitation is very weak. So since there is no excitation or no absorption here, there will be no emission. So there will be no emission here. There will be no emission of 680 nanometer emission. So basically here, in this case, the emission is all contributed from Psi 3. However, after hybridization, after conditions that inducing the hybridization between these two oligonucleotides, so you can see now the two oligonucleotides will form duplex. So this duplex formation will bring the Psi 3 and Psi 5 to very close distance. So their distance will be less than 10 nanometer. So there could be a frat occur between Psi 3 and Psi 5. Now, if we shine 540 nanometer light, so again, Psi 3 will be excited to generate 590 nanometer emission. But in addition to this, there will be small amount of energy will be directly passed from Psi 3 to Psi 5. So this energy is from the excitation of 540 nanometer light. So part of energy will be used to generate emission, and the very small part of it will be converted and directly passed to Psi 5. So this is a frat. It's a radiationless energy transfer. So Psi 5 will use this energy to be excited to the excited stage. Then it generates an emission photon. So this is the emission light. So the emission light will be characteristic of Psi 5 emission light. It will be 680 nanometer emission. So this is the frat. So we can see that the excitation light excites the donor, and the donor passes energy by radiationless energy transfer to the acceptor. Then the acceptor is excited, then generate acceptor's characteristic emission light. So the application of frat is very wide in cell biology. So it could be used in real time to monitor the distance, and then most important, to monitor the interaction between two molecules. So you might wonder why the distance means interaction. This is because when the two molecules, their distance are less than 10 nanometer for a long period of time, that means they are interacting physically. Here is a further example to show by using frat to detect protein interaction. So this is a protein X. Protein X is fused to blue fluorescence protein, BFP. And this is the protein Y. protein Y is fused to green fluorescence protein. So when there's no protein interaction between X and Y, the BFP and GAP are further away from each other, there will be no frat. So under this circumstance, if we use violet light to shine this system, there will be only blue light generated because there's no frat between BFP and GAP. Well, under the protein interaction condition, so when X interacts with Y, there will be frat between BFP and GAP because the distance between the two usually is less than 10 nanometers for protein interaction. So here, if we use the same intensity of violet light to excite the BFP, of course the BFP will still generate the blue light as the emission light, but however, the blue light will be reduced because part of excitation energy will be used to be directly transferred to the YFP. So then the YFP generates its characteristic green light. So in this system, when X interacts with Y, when shining the violet light, there will be green light coming out. So because the frat between BFP and GFP. Therefore, we can use the occurrence of frat to detect protein-protein interaction. The best way to quantify frat is by using FLIM. FLIM stands for Fluorescence Lifetime Imaging Microscopy. FLIM measures the fluorescence lifetime. So recall from lecture 2 that we have used Jablonski diagram to explain molecularly how does excitation work. So that the photon is absorbed by the fluorophore, and fluorophore jumped from the ground state to higher energy state, excited state. So it stays in the excited state for a certain time, and this time is the fluorescence lifetime. Then it emits a fluorescence photon and return back to the ground state. So the fluorescence lifetime is the residual time that a molecule which has absorbed a photon remains in the excited state before returning to the ground state level by re-emitting a photon. So the fluorescence lifetime usually is in the range of nanoseconds. This is a very short time. That's why when we stop excitation light shining of our fluorophore, we can immediately see there is no emission light coming out from the fluorophore. But however, there's a delay of nanoseconds between the two. Usually we don't notice it. So the fluorescence lifetime is sensitive to frat. So that's why we can use fluorescence lifetime to measure quantitatively the frat. But fluorescence lifetime is independent of the concentration. So that means the concentration high or low for particular fluorophore, it has same fluorescence lifetime. So this is different from fluorescence intensity. Fluorescence intensity is proportional to the fluorophore's concentration. The higher the concentration, the higher the fluorescence intensity. FLIM is commonly used to quantitatively measure frat. So here is an analogy to help us to understand why the fluorescence lifetime is sensitive to frat. So here is the ground state. It's like a lower level water reservoir. And here is an excited state. It's higher level water reservoir. So the molecules jump from ground state to excited state, and it does not stay forever at the excited state. There is a radiative decay to drain the fluorophore from excited state back to the ground state. So this radiative decay, for example, is by fluorescence. By generating an emission photon, the fluorophore will quickly return to the ground state. The frat is a type of non-radiative decay. So once this fluorophore establishes a frat with acceptor fluorophore. This will make the excited state to return to the ground state faster. So here you have one tap to drain it. The frat is as if another tap. So by the action of 2 taps, it drains the excited states much faster. So this is why frat makes the fluorescence lifetime shorter. So because this water will be drained faster than this case without frat. So flame actually is the most rigorous method for determining the frat. So generally we use flame to quantitatively measure the strength of frat to determine whether there is a protein-protein interaction in cell biology. So in summary, during frat, the donor fluorescence lifetime reduces. So the flame and frat have very wide application in cell biology. So now let's give you an example. So here we are going to use GAP and M-cherry for a frat pair. So GAP is green fluorescence and M-cherry is red fluorescence. So they could be a frat pair. So now let's show you the spectrum. So here is the spectrum of GAP. This is excitation spectrum, emission spectrum. So here is the red fluorescence protein M-cherry, excitation and emission spectra. So we can see that 3 criteria must be fulfilled. So the first, the donor, so GAP here is the donor because its excitation and emission is all shorter wavelengths compared to red fluorescence protein M-cherry. So the donor's emission spectrum must overlap with the acceptor's excitation spectrum. So here is overlapping between the two. And if their distance is less than 10 nanometer and their dipoles could orientate it approximately parallel, then there could be a frat. So in our case, HeLa cells are simultaneously transfected by GAP and M-cherry coding sequences. So you can see that GAP expresses and M-cherry also expresses within the same cell. So this is a GAP channel, this is the M-cherry channel. So both GAP and M-cherry are in a very diluted solution inside the cell. So normally their distance is far away from 10 nanometers. So there is no frat between GAP and M-cherry because distance criteria is not fulfilled. So we can use a flame to measure the fluorescence lifetime. So there is a microscope specifically designed for flame. So we're not going to cover its operating principle here. So here is a flame map. So that is usually color coded. So for example, in this as a heat map. So the blue means 2.4 nanometer and green is something in between 1.8 and 2.4. Well, the red means 1.8 nanometer. So here from this color map, this image, we can see that most of GAP, the donor fluorescence lifetime, is about 2.4 nanometer. So now we can make a chimeric protein. We can use recombinant DNA technology to fuse GAP coding sequence with M-cherry coding sequence by using 7 amino acids to link the two. So when we transfect this chimeric protein inside the cell and the cell express this chimeric protein, so this GAP and M-cherry are now linked by 7 amino acids. So the distance between GAP and M-cherry are very short, it's less than 10 nanometer. So there could be a possibility for GAP and M-cherry to have frat. So here is a GAP fluorescence channel. There's GAP fluorescence, there's M-cherry fluorescence, and there's also frat. So recall that frat is independent of the concentration. So it doesn't matter this molecule, this chimeric high or low, they all generate the same tau map, same fluorescence lifetime map. So this is also color-coded, just as this one. So we can see from this map, this image, we can see that the majority of molecules here shows yellow. Yellow here means around 2 nanometers. So now the fluorescence lifetime now is, instead of 2.4, now is 2. It's lower than 2.4. So that indicates there is a frat between GAP and M cherry in this case. So after establishing GAP and M cherry as a valid frat pair, we could use frat to study the chromatin compaction. So here we use histone 2b fused to GAP and histone 2b fused to M cherry. So both transfected to the cell, and the cell expressing both. So the GAP or M cherry are incorporated loosely into nucleosome, because histone 2b is one subunit of nucleosome. So here is a chromatin. So this is a loose chromatin. So this is nucleosome, these are DNA. So here some nucleosome are labeled by GAP, some are labeled by M cherry linked histone 2b. So this is all stochastic. So under loose chromatin condition, because the labeling is at a low density, so they are relatively far away. They're far further away than 10 nanometer. So there's no frat or low frat. And during high compactation, highly compacted chromatin, so when the chromatin is highly packed together, so it's these nucleosomes are physically near each other. So the GAP and M cherry could be physically near each other, and some of them will be less than 10 nanometer. So therefore there could be a frat occurring between this GAP and M cherry. So here is the experiment. So here is the nucleus, this is nucleus under ATP sufficiency condition, and here is the nuclear under ATP depletion condition. We know that within the nucleus there is chromatin. So here the color code shows the frat efficiency. So here we can see that mainly is green. Green indicates a low frat efficiency. That means GAP and M cherry have less frat in this condition. So this corresponds to loose chromatin. Well, in this condition, ATP depletion condition, the frat efficiency increased. So it shows red. So red corresponds to higher frat efficiency than green. So this corresponds to a compacted chromatin. So by using frat, we can very conveniently monitor the chromatin compaction in live cells. So if you are interested, you can go ahead to read the reference in the reference list. So this is the end of today's lecture.